And so last time, we gave a precise definition of what an alignment is between two sequences. We gave a precise definition, although a fairly simple one, of what we mean by two sequences being similar. And I remind you that it ended up as an optimization question. Of two sequences, S1 and S2, was defined as the maximum value you can possibly get, the maximum over all alignments of S1 and S2. Over all possible alignments of these two, you wanted to maximize the number of matches that resulted minus the number of mismatches minus the number of spaces. And this value, the largest number that you can possibly get over all alignments of S1 and S2, was defined as the similarity or similarity value of S1 and S2. And so it gives you a way of, a numerical way of talking about how similar two sequences are. And associated with that value is an actual alignment, which shows you the relationship between S1 and S2 that has that value. Okay. The next thing we did last time was to ask how many alignments are there? Because if there aren't very many, then you can write a very straightforward computer program that generates all of them and calculates this number, and then just takes the largest over all of those. Ultimately, we want a method, and it has to be efficient. It has to be something we can actually use in practice that will calculate this number. And what I was pointing out last time is the number of alignments is much too large for a straightforward approach, a complete enumeration approach, to be practical. It's totally out of the question. And in fact, one of the things you have on your homework this week is to run a program, two programs actually, that calculate the number of alignments. And you'll see how rapidly that grows. And in the notes, the class notes that I put on the web from last lecture, I actually have a kind of approximation. Last time I gave you an exact formula. But we can approximate what its value is, and you'll see that it has exponential growth, very rapid, explosive growth, as the lengths of the strings increase, lengths of the sequences increase. And you can see some of those first values, what their actual numbers are with the programs that are available on the web. So I'm not going to say anything more about this, except to say that any approach based on enumerating all the alignments is totally impractical. Now, what I really should do at this point is start talking about how we efficiently compute this without complete enumeration. And I may get to that today as well. But because the lab, we have this synchronization problem with the labs, the lab started on the very first day of class, so it's slightly running ahead of the lectures. I want to say enough today about things that are related to the lab that you can actually jump into the lab. So I'm going to skip over this topic of how to compute this one and do this slightly out of order and talk about richer models right now. It's something that would otherwise come later. Because this week's exercise involving Xperal is to explore different models and explore different parameters of different models to see which ones give you alignments that seem to have biological meaning. And the way we're getting at that is in two different exercises where we have the alignment that's believed to be right, okay, believed by the appropriate biologist to be the alignment that exposes the biological meaning. In one case, that alignment came from crystallographic studies. You have two proteins. You know what their three-dimensional structure is. And then you can do some kind of superposition of those three-dimensional structures. And that implies an alignment of the sequences. Places where the structures line up pretty well, excuse me, are places where you want the alignment to line up pretty well as well. And certain properties secondary structure, the alpha and beta structures of those proteins, you'd like those to line up properly as well. So in the Barton data, you start with an alignment that is believed to be correct. You input those sequences to Xperal without the alignment, and then you see what combinations of models and parameters gives you back the alignment that Barton thought was right. And what's most instructive also is to see that with many other choices, and there's lots of other choices, you get alignments that are really crummy. And that should be kind of a warning in general about how to use this technology. The other thing that you get when the parameters are wrong, the model is wrong, is you get lots of equally good alignments. Okay, alignments that the system, in terms of this objective function, you see, ties are possible. There's nothing in this that says there's only one alignment that gives you this biggest value. There certainly could be more than one alignment that does that. And if you have a set of parameters where you get thousands or tens of thousands of alignments that give you all the same largest value, then this is not a technology that's going to be of great use. How do you know which one of those alignments is the one you ought to run with? So that's another thing that you're exploring in Xperal. And if you get lots of polygons, that's probably not a good thing. In fact, what I'm doing now is I'm telling you the answers to the questions on this week's lab, but you have to connect the answer to the question properly and make that make sense. And I'm going to show you a little bit more about Xperal in a minute. So richer models. So how do we want to enrich this model? The first thing, actually, and this relates to the second exercise, in the early 1980s, this was sort of the model that people were playing with, alignments under this objective function. But they started differentially weighting mismatches and spaces. So that's modeled by putting a multiplier x and a multiplier y here, and then saying, well, I think x should be, a half, and I think y should be 12 or whatever. Now, of course, the case that we've looked at so far, x and y are just equal to 1. But if you think that mismatches happen more frequently in nature than spaces do, then maybe you should make this y bigger than x. You should put a higher penalty for having spaces so that the alignment that gives you this maximum value is one that has fewer spaces and maybe more mismatches. Okay, so having these multipliers, then you have to argue what the proper multiplier is. So that's the first way in which people enriched the models. Multipliers on mismatch and space penalties. Okay? You might ask why I don't put something on matches here. Why x, y, z? Why don't I play around with the value of a match? What's the answer to that? Yeah, you can conceptually think about saying, yes, this is really a parameter here, here's a parameter. There are really 3 parameters. But for any choices of x, y, and z, if I just divide here by z, then this one goes back to being 1. This is x over z. This is y over z. And that's just a different, since I'm going to let this parameter and this parameter run through its all possible ranges, it really doesn't matter whether I call it x divided by z or just call it x. Okay, so another slightly less clumsy way of saying this is there are really only 2 independent parameters here. if I have 3, I can think conceptually having 3, but there's really only 2 that are independent. I get the full range of behavior by setting this to 1 anyway. And then from a practical standpoint, it's good to only have 2 parameters because on a computer screen, you only have 2 dimensions. There are actually programs that will show this kind of thing in 3 dimensions, or at least a 3-dimensional representation. But you don't need to when you only have matches, mismatches, and spaces. 2 parameters are enough. Okay, so multipliers on mismatch. That's another way people got to richer models that hopefully found alignments that were more meaningful from a biological standpoint. And part of your exercise this week has you playing around with this model, okay, without any further embellishments. Now, what I think Fitch did in the in the 1980s somewhere is he had, there wasn't a lot of data then, but he had 2 fairly short sequences where he felt that he knew what the right alignment was. I mean, the alignment that meant something biologically. And he wanted to see how good is this technology, this alignment business that people were all starting to do. And now we just do, massively. And what he did was he took his example, his 2 sequences. Let me just clean this up a little bit. Number of mismatches minus number of spaces. And, yeah, he multiplied by x, multiplied by a y. And by hand, he did what Xperal does now automatically. He computed this nice decomposition of xy space where he could see all of the possible alignments, all of the alignments that maximized this objective function over all choices of x and y. Whenever you fix a particular value of x and y, then you have a specific optimization question, and you can find through the method I haven't yet told you about, but you can find the alignment that maximizes that objective. And he did that for all x and y in a systematic way that Xperal now does automatically. And he looked at all those alignments, and he found that none of them were really satisfactory in terms of capturing the alignment that he thought was right, that he thought was biologically relevant. And that led him, and others, he wasn't the only person, into this notion of gaps. So that's the second way that this model is enriched, particularly for protein. Okay, so what is a gap? Here's a definition. A gap is a contiguous sequence or interval of spaces. Okay, so if I have, let's say, A against A, and then 3 spaces here, Bx, Y, Z, Z, W, P. Okay. This is an alignment that has however many matches it has, however many mismatches it has, but it has 1, 2, 3, 4, 5, 6 spaces, and only 4 gaps. No, sorry, 3 gaps. So the 6 spaces are organized into 3 gaps. And 1 gap here, 1 gap here, even though this is just one, this is just a single space, it's nonetheless a gap. And here's a longer gap. So you can have different alignments that have exactly the same number of matches, mismatches, and spaces, but with different numbers of gaps. The spaces are organized into different number of gaps. And the way they're organized into intervals can differ. And what he was asserting, particularly in protein, is that an alignment that's sprinkles the spaces all over the place doesn't look very real, doesn't reflect the biology that he knows, doesn't reflect how two sequences that are related to each other by biological processes ought to look when you do the best possible alignment. In his view, they should have just a few gaps. They may be very long, or they may be very short, but the spaces should typically be organized into runs or intervals of gaps. So he then, remember, he looked at this objective function in completion. So I'm moving back over here now if you want to. Thank you. He looked at this model. This is a model. It has no, this is a mathematical model associated with a algorithmic problem. But it has no notion of a gap in there. Even though it has x and y, it has no notion of a gap. It can multiply mismatches in spaces, so he has a way of fine-tuning that. But even with all the fine-tuning that he did, he couldn't get an alignment that recreated the alignment that he thought was meaningful. And so he and others, at that point, introduced into the objective function this further term, which is subtract off something for gaps. Okay? You don't want to have a lot of gaps. So we're trying to maximize, we're trying to make this big. You want a lot of matches. You don't really want a lot of mismatches. You don't really want a lot of spaces. But you'd also like to control how many gaps you have. Okay? And he was, at the time, wearing that T-shirt. Would you stand up and... yes. No. Yeah, you know what T-shirt you're wearing. And face the camera. And anyway, the camera should capture that one. I guess the camera's not listening. Anyway, he thought it was important to mind the gap. Which is what that T-shirt says. And how many people know where that comes from, by the way? Just an odd question. Only one or two? Yeah, it comes from the underground in London, where they have it as you get off the train. It's stenciled there almost every exit, so you don't fall through or fall too far. Anyway, they sell those T-shirts everywhere. So if you're ever in London, pick up one. So anyway, they introduced the notion of gaps. So you don't want to have a lot of gaps, and you can control things to some extent by including this in here. Well, the next obvious step is gaps and a multiplier on the number of gaps. So now we have x, y, and z. How did I get my y inside there? That was silly. Y times the number of spaces. So z times the number of gaps. So now that we have four elements of an objective function, we have matches, mismatches, spaces, and gaps, now we do have three independent variables. And we would like to have a multiplier x, y, and z. And that's why some parametric programs actually would like to display this in 3 dimensions, showing axes for x, y, and z. Xperal only shows things in 2 dimensions, and you have in your menu, your choices, you have to decide which 2 out of these 4 you're going to parameterize. Okay. So what Fitch next did is he parameterized these 2, I believe. But you can see in the notes y and z. Just forget x because that used to go there. And by playing with y and z, and for each setting of y and z finding the best alignment, the alignment that maximizes the subjective function, he was able to find an alignment that recreated the alignment that he thought was right. And so in your homework, in your exercise this week, you should be able to do that as well. And the same thing with the Barton data. That's more complex. It's a much longer sequence, but you should be able, after failing to recreate the alignment that Barton starts with, or by many settings and different models that seem to give you too many choices and so on, you should eventually, with the right model, be able to get an alignment that seems right in comparison to the Barton's alignment. Okay, but actually for the Barton, there's a third very important element that makes richer models, and that is substitution. Substitution, I've forgotten what we call them in Xperal, but generally we call these substitution matrices. I think in Xperal it's called something else. But anyway. So substitution matrices, I mentioned this briefly today in lab, and we'll talk about this more throughout the quarter because there's a question of where do these things come from? How do you get good substitution matrices? But what this is saying is that not all mismatches, and in fact not all matches, are of equal significance. So a mismatch, an alignment that matches, for example, isoleucine to leucine, is not a huge, you don't have to invent a huge, difficult biological story for how that happened. You can still imagine that two sequences that have this kind of a mismatch are highly related, because these are two amino acids that are quite similar and that mutate one to the other fairly easily. And therefore, this kind of mismatch should be less penalized. Should be less penalized than a mismatch, say, of tyrosine. Is this tyrosine? And what? Tyrosine is a Y. Okay. And what's a G? Glycine. So this is big and this is small. And there are other ways that you can find two amino acids. One's charged, one's not, and so on. Hydrophobic, hydrophilic, etc. Two that are quite different, where a mismatch between those is postulating a large chemical and biological change, and therefore probably should be penalized much more than this kind of a mutation. And so what you have are substitution matrices that encode all of those. So you just have, this is particularly true for amino acids, but also true in terms of DNA as well. So you would have all of the amino acids and so on, the 20 here and 20 here. These are the identities along here. And some value for particular mismatches that would be found in this matrix. Okay? And even some value for matches that are found in the matrix. Now, generally, the values for matches are going to be positive, and the values for mismatches are going to be negative. I should have told you exactly how this is used. When you have an alignment, the score you give for that alignment is no longer just the number of matches. It doesn't start out just with the number of matches, but it is the score that's taken from the summation of the values that are taken from this matrix corresponding to the matches that you actually have in there. So over here, well, this is not an amino acid because B is not. Maybe there's some other ways in which this is. Anyway, just imagine it was. So you have a match of A to A, and you'd go into the substitution matrix, and you'd say, you'd find out what value does an A match have. So that contributes to the value for a match. What do you get from a Z? What do you get from a P? And so on. And those are taken from that particular substitution matrix. And then mismatches you would also go into the substitution matrix and you'd find the QR value. And that would then be taken, if mismatches are negative, that's actually as a plus. And in fact, if you look at Xperal, one of the windows, the objective function windows, has number of matches plus number of mismatches. And then it has a subtraction for spaces. And I think I mumbled something in the notes about how that's kind of an error. But really what it is just, it reflects the fact that the values from mismatches in the matrices are already negative. And so we're really just, from an arithmetic standpoint, we are adding those. We don't want to subtract a negative because then it'll become positive. But the spaces still are subtracted off. Okay, so the substitution matrices are very, very critical in terms of getting the right alignments, getting alignments that are biologically meaningful. And again, this week's exercise is to get you to sort of discover how important all of the pieces of the model are in order to get a good alignment, a biologically meaningful alignment. There are many substitution matrices that have been proposed out there. The one that you're using in the exercise is something called PAM 250 plus 8. And it's in a file that's in the directory that you're working in. You can actually just look in that file and you will see what the values are. You have the two coordinates on that array that's in there, and you can see what the values are. Now, the PAM matrices we'll talk about later, how they were constructed. It's a whole series of matrices, and this sort of indicates which PAM matrix it is. The plus 8 means they took the original PAM matrix and they added 8 to all values. A little bit of alchemy. But today, and PAM 250 used to be the default matrix that a lot of people would use when they didn't know any other one that was better. Today, this is a little bit dated. And people tend to use the Blossom series of matrices, which we'll talk about also later in the quarter, where these things come from. But I don't have that built in. You can use it. I'll make it available. And in Xperal, when it asks for what scoring matrix, there is an option for other. And as long as you have it in that directory in the right form, you can use it. So you can try that out if you like. Blossom 62, I think, is now sort of the default. When you don't know anything else to use, people tend to use that one. But we'll talk more about these. I just want to say that, particularly for protein comparisons, DNA is somewhat different. But for protein comparisons, all of these features of the model are really have ended up being critical in terms of reliably getting back good alignments. And probably the most important are in opposite order here from the way I've done it. The substitution matrix is really important. Gaps in the model tend to be significant and very important. And then the actual multipliers more fine-tune things. Although, now let me just tell you some terminology, and then I'll tell you the although. When you're looking at protein under this model, gaps, the gap Z is oftentimes called the gap initiation cost. Okay, and you'll see that referred to in the Xperal notes, and you'll see that in biological literature. So that is what it costs you for a gap. So if you think about starting a gap and where you don't know how long it's going to be, but there's going to be a gap, then you can think about initiating a gap without knowing how long it is. Okay? So that's why it's called a gap initiation. What you pay for a single gap is z here. And whatever you set z to is the specific gap initiation. And then y is referred to as gap extension cost, or penalty, cost penalty. Extension cost. Okay? And again, if you think about this, when you start a gap, you have to pay to start it, but you don't know how long it is. Every time you add a space to it, you're extending it. And you have to pay y for each one of those spaces. So that's called gap extension. Okay? I think it's simpler just to say there's a cost for the spaces and a cost for the gap. But historically, those two terms have come up, gap extension for spaces and gap initiation for cost. Now, the although is that even though people don't always play around with y and z the way Xperal allows you to, there are definitely beliefs about the rule of thumb out there for how y and z ought to relate to each other. And some of these have become almost gospel. But if you go back to where the value, the advised value of y and z came from, in many cases it came from somebody sort of off the top of their head. They said, let's try y of 1 and z of 12. Something like that. And then it gets written down on some paper that those are the right two values. And then it becomes part of somebody's program and the manual that's associated with that program. And pretty soon it's as though there was some really deep analysis that gave you that. So generally, the gap initiation should be more expensive. It should be more difficult to start a gap than to extend it. And actually, the 12 to 1 is actually some default values for some alignment systems that are out there. But in Xperal, you don't have to rely on that just single choice. You get to play around with what you think y and z ought to be. And in comparing proteins, these tend to be the two parameters to play with. What you have on spaces and what you have on gaps. Okay? So I want to show you a little bit more now about Xperal specifically. And this whole business is called parametric alignment because we have parameters on portions of the model. Now, how well is that coming out in the TV version? Can you make that fill the whole space? See, I've noticed that. And try to play whatever you can so this is most visible on the TV screen. Thank you. Okay, so here's my the objective function. We're trying to maximize the value of the matches, and I say value now because that is implying we're taking the summed value from some substitution matrix, okay, minus the value of the mismatches. Well, again, whether this is a plus or minus really depends on what's the actual shape of the what are the values in the substitution matrix. But you should all understand that mismatches are bad and somehow they should be penalized, whether the value in the substitution matrix is written there negative which case you add here, or whether it's in there positive, in which case you subtract. You just have to be careful about what you're doing. Minus the number of spaces, but I'm going to have a multiplier alpha. Well, that used to, over here that was a y. Okay, now it became alpha. We all speak Greek now. Minus beta times the number of gaps. Okay? And so we have two parameters, and that gives us a alpha beta space for our choice of what alpha should be and what beta should be. The default in some systems has alpha of 1 and beta of 12, but in Xperal you get to explore the entire range of values in these two parameters in order to see what's the consequence of setting the values differently. And what do you get out of Xperal is you get a decomposition of the alpha-beta space into these regions, which are called convex polygons. And you can prove, it's not something we're going to do in this class, but one can prove that this decomposition has the following property, that whenever you pick a value for alpha and beta inside of a particular polygon, no matter what choices you make for alpha and beta, if they fall within the same polygon, then the same set of alignments are optimal for either point. Saying that slightly different, if I pick this point, for example, right here, and I find an optimal alignment for that choice of alpha and beta, the same alignment will be optimal for that choice of alpha and beta. There may be ties. There may be more than one alignment that's optimal at this point, but that other one, all of them that are optimal over here will be optimal over there. Okay, by optimal, I mean they maximize. They make this maximization function as big as possible. So the polygon, inside of a polygon, it doesn't really matter what your choice of alpha and beta is. You're going to get back from the alignment algorithms the alignment that maximizes the objective function will be the same one anywhere in here. Also, the converse is true, that outside of that polygon, no alignment that's optimal for a point inside the polygon will be optimal anywhere outside the polygon. Okay? So if I pick an alpha and beta here, and I find an alignment that maximized the objective function for that alpha beta, I know for sure that that one will not be optimal over there in any other polygon. Okay? So what this decomposition shows you is exactly where the values of alpha and beta cause a real change. Okay? Inside one single polygon, you have a collection of alignments that are optimal. They're optimal nowhere else. And so this decomposition tells you how the optimal alignments behave as a function of alpha and beta. That's what Xperal does for you. Now, let me show you an actual picture. This actually is a little bit, this one is taken from a earlier version of Xperal. So what's up on all the choices up on the top are not quite the same as what you're going to have in your exercise. But all the important ones are there. And this is actually the final decomposition that you should end up seeing. And for the Barton data. And let me show you the most important places to look. One of them is right here. And you can see that, well, there's one polygon that's shaded. And the alignment that's associated with the shaded polygon is shown up in the alignment window. So we're seeing up there an alignment. It's got some, you know it's an alignment because it's got some spaces in it. It's got some gaps in it. And here in this window that says VAR, it tells you how many matches that alignment has. If you can read this, it's 29. 29 matches. The next one is how many mismatches. That's 73. The next one is how many spaces. It's 16. And the next one is how many gaps. That's 5. Okay? So in the alignment that's shown, which is optimal somewhere, well, it's optimal everywhere inside this polygon, you have those features, 29 matches. Yeah?
You said all the interior points within a polygon are as optimal as the other points. But what about the lines?
Okay, so the question is, okay, what I said was that for any alpha, beta point or xy point inside of a polygon, any two of them, the same alignment that's optimal at one point will be optimal at any other point. And the question is what happens along a border between two polygons. There, all of the alignments that are optimal in the interior here, and all of the alignments that are optimal in the interior on the other bordering polygon are both optimal along this border. And similarly at a point, that's the intersection of more than two polygons, all of the alignments that are optimal in the interior of the polygons are optimal at that point. So you have a lot of different co-optimal alignments. Okay, so when you look at an Xperal display, you want to see what optimization did you choose. This is one that's called n-gaps free. I haven't explained to you. I should tell you what the terminology is. That what we've seen so far, those alignments are called global alignments. Global because every character in the two sequences participates in giving the value to the alignment. Sometimes there's a belief that gaps that are at the ends of the alignments should not be scored. If you have one sequence much, much smaller than another sequence, for example, you're going to have very big gaps, probably, at the ends. And those are not necessarily meaningful. So there's a variant called n-gaps free. And Xperal gives you several choices. Another one's called local alignment, which we'll look at a lot in this class. At any rate, you'll see in your lab notes which variant to use. But here is where it shows what variant you're using. This shows the numbers of matches, etc., of the alignments that are in this polygon. Over here, what this shows is, remember, the Barton data, you input aligned data. You input aligned sequences. In the file, and you can look in that file, the Barton Go file, the two sequences are actually there as an alignment. There are spaces in them. That's the alignment that Barton thought was the right one from the basis of crystallographic studies. So we know how many matches, mismatches, spaces, and gaps that alignment has. So what's shown over here is the number of matches, mismatches, indels, and gaps that alignment has. It turns out that one in the Barton data has 30 matches, 72 mismatches, 16 spaces, and 7 gaps. Okay? The alignment we're seeing over here has 29 matches instead of 30, has 72 mismatches instead of 72, has 16 spaces, which is the same, and is organized into 5 gaps, whereas this is organized into 7. The alignments that are in here may not be the closest one, the most similar one to what was input here. In the current version, not on this one here, but in the one that you're playing with, there's actually a button that says, find the one that's closest. Forget about showing me the full decomposition. It just works until it finds one that it knows is the closest, and it stops there. But you can always sort of compare how the alignment that's shown here compares to what was input, if you have an input alignment. In the Fitch case, you're going to type in the sequences. There's no alignment given. Then what's down here actually tells you some numerical differences between the alignment at hand here and the input alignment. It says at point x, y, and I'm not going to tell you how it picks which points to look at, but at point x of 0 and y of 5, it picks a particular point. The value of the alignment shown up here, which is in here, well, this 0, 5 should be inside 0, 5. Yes, sorry, 0 is x, and 5 is along here. 0, 5 should be in there. At that point, the value of the alignment that's shown up there is 941. But the value of the input alignment is 932, which is a numerical difference of 9, which is like 1%. Okay? So that's another reflection of the closeness of these two alignments, that this is a very small percentage difference between the two. Now, another thing that we have in the current version of Xperal, it's not in this slide, is something that's up here, which shows you the number of equally good alignments in each polygon. So in this polygon, I vaguely remember there's only one alignment that gives you the maximum value. It's optimal every point inside here, but there's only one of them, or it's 18. I don't remember which. Some of these in here vary between 1 and 18. Okay? And then these buttons up here say next and previous, and you can scroll through the alignments. Sometimes the changes are quite subtle, but it'll show you the different alignments that are called co-optimal. And if there are more than 10,000 of them, it just says more than 10,000. And part of your homework exercise is to think about what does it mean if you've picked a model and a choice of weights where you're in a polygon where there are 10,000 equally good alignments. What does that mean about the value of this alignment as a tool to find biologically meaningful alignments? If there are 10,000 that are equally good under the criteria that you're working with, it's not a very precise tool. If you have only one, it may be the wrong alignment, but at least it's only giving you a few choices. Similarly, the question of how many polygons there are is the same issue. If there are too many polygons, then there's too much to explore. And then you can be a little more precise. If there are 18 in here, you can ask, how do they really differ?